Thank you for the invitation to present some of this work. So I'm going to present some published work from last year, which was largely done by a couple of students. Hang on, let's see if I can make this long. Which was done by a couple of students in my lab, Harry Sutton, now at La Jolla. I tried to get him to give this talk, but he was giving another talk today, so he's left it to me to do, helped by Andy Gow. And then this is based on models built by Hayley McNamara in the lab, now in Germany. And because it's a published story, I'll try and give a little bit of, I'll try and tell you a bit more of the background and some of the things that may not come out were you to read, what's necessarily a very short and abbreviated paper. My background is actually not really as an immunologist. I'm more of a parasitologist by background. People in my lab would call themselves immunologists. And we work on the malaria parasite, which is, the Plasmodium parasite, which causes the disease malaria. Malaria, the disease, is largely caused by this sort of blood stage, where you have an asexual infection of blood stages leading to a massive increase of parasites and anemia and cerebral malaria. But our vaccines are mainly targeted at these things called sporozoites injected by the mosquito. And the reason for that is because it was shown that if you immunize with irradiated sporozoites, you can get sterile protection against malaria. And a lot of that protection is antibody-based protection targeting this repeat region of a surface molecule called the circumsporozoite protein. And when I started my lab 10 years ago, we had this question, which was, given that this is a highly repetitive antigen, could it be inducing suboptimal immune responses? And mostly that's turned out to be not true. We found that you would get efficient affinity maturation, class switching in response to this. It's largely a T-dependent response. But in order to sort of ask those questions in the first place, one thing we did was to develop this BCR knock-in mouse system to track these CSP-specific B cells. We now have a large number of human monoclonal antibodies, highly potent human monoclonal antibodies, some of which are actually in clinical trial as malaria prophylaxis. But when I was starting the lab and doing this work, we had a slightly lousy mouse antibody called 2A10. And we solved the structure of this antibody and looked at its binding with CSP, but we also made mice based on this antibody in order to try and track B cell responses to Plasmodium. And what we did was we knocked in the heavy chain of the 2A10 antibody. We tried to make a mouse with a light chain as well, but that light chain knock-in mouse never actually really worked terribly well. So we ended up with this mouse which just had the heavy chain knocked in. But one thing we did was we inferred the germline of that heavy chain, and we made the mouse using the germline sequence so that everything would have physiological affinity. And in this mouse, what you find is that around 1 to 2% of the B cells bind to the antigen. It's 1 to 2%, I guess, because you need the right light chain there. And as I say, we never managed to fix the light chain. That actually turned out to be an advantage in the long run, because if you actually look at these cells and you stain them with probe and you put that against IgM or CD1 or CD79, some kind of BCR thing, you can actually distinguish these different populations that have high, intermediate, and low affinity for antigen. And we guessed at that time that this was due to the use of different light chains. And sure enough, these different populations, actually, they're not very different light chains at all. They all use this kappa 1094 light chain, but they pair it with different J's, and that results in the presence of different aromatic amino acids in the light chain CDR3. So we kind of have this mouse right away, which has these 3 different populations that we can track, and we asked whether this could make a difference to whether cells could become. We were interested in this question of whether cells could become plasma cells or germinal center cells, as it was always thought that high affinity cells typically became plasma cells. And a second thing we did, or that Harry did in the lab, was he spent a long time sort of characterizing the pattern of affinity maturation in this lineage. And he started this project, back in 2015 as an honors student, as an undergraduate student in the lab. And every time he'd do an experiment, the technology would move on and we would want to use a different kind of single cell approach. So this was done with a very traditional initially this was done with kind of a SMART-seq2 approach, and he was actually sequencing single B cells, and he was amplifying up the light chain sequences. And if you sort of follow these sequences over time, at day 4, you have no mutation. By day 7, you see a mutation in the CDR3 of the light chain, which has swept through by day 8. And then you see a number of other mutations emerging over time. Notably, there's a glutamine mutation at position 106. So the light chain CDR3 looks like this. It has this leucine to phenylalanine that basically appears, on the morning of day 7, and by the afternoon of day 7 has basically swept through the entire germinal center. And then these additional mutations that evolve over time. Now, I try and tell, and then this is kind of a schematic of how you can see everything is unmutated. And we can track whether these cells come with the J1 or the J2, so whether they're intermediate affinity or high affinity cells in their sort of germline, depending on exactly which light chain they have. And what you can see is, of course, everything is pretty much germline at day 4, but as time goes on, you get this accumulation of mutations sweeping through the germinal center. We also, of course, and Hannah in the lab also prepared recombinant antibodies of all these different variants and formally showed that these cells actually, or these variants lead to increased affinity. Notably, even though this J1 variant binds the tetramer really quite nicely, when you actually run it on a kind of an octet analysis, then you actually find that you can't actually detect the affinity. Or you can see a signal, but you can't assign an affinity to it. So I try and tell my lab that, what you need to do is you come up with the questions and you build the tools afterwards. But in this particular case, the tool was so compellingly lent it to a specific question that that we felt that we had to go after this question. The question this tool seemed to lend itself to was the question of what determines cell fate decisions in germinal centers. And this was always, I think, the traditional model. People noticed that plasma cells have much higher affinity than memory cells, and so they infer that in the germinal center, low affinity cells become memory cells, and I think that's now well established, but that high affinity cells would preferentially go and become plasma cells. Mark Schlomchik proposed a different model, which was that memory cells emerged early when the overall affinity of the germinal center was low, but plasma cells emerged late when the affinity is generally higher. And we thought that we could exploit our canonical pattern of mutation to distinguish these two possibilities. So the experiment was this, which was to put in our 2A10 cells, so our CSP-specific B cells, immunize them, in this case with sporozoites, so Plasmodium parasites expressing falciparum CSP. And then at day 7 and at day 21, when we kind of see these two canonical mutations, this leucine to phenylalanine and then this glutamine mutation, and ask basically, can we see these mutations? And if we can see these mutations, are they preferentially in any population of cells that looks like it's becoming a plasma cell, or are they not in cells that become memory cells? And so we did this simultaneous VDJ and transcriptomic sequencing, which of course other people have talked about many times today. We did multiple mice. We took advantage of hashtagging in order to get replicates and therefore to get some statistical power in our analysis. And we also used barcoded antibodies at the same time for markers that other people had inferred were pre-plasma cell markers. So this this is a little small, and I apologize, but the take-home message here we've got 5 different mice. This is at day 21. This is essentially the pattern of mutations across the heavy chain and the light chain. And I think what was so striking to us was, and it's based on around 500 to around 1,500 cells per mouse, depending on how many came out with the hashtagging. But what you can see is that every mouse essentially is showing exactly the same pattern of mutations. You can see that leucine to phenylalanine down here. But then you can also see now a number of additional mutations that pop up around, particularly around the CDR1 and the CDR2 of the heavy chain. Another thing we were able to do was to ask whether if I have one mutation, am I more likely to have another mutation? We were concerned that there might be different pathways to high affinity other than the ones that we originally found through these light chain CDR3 mutations. And what you do find is that, well, everything has the light chain 114 mutation, the leucine to phenylalanine, and then half of things have this light chain 106. But once you get that additional mutation, you're predisposed to get more mutations on top of that. And they go in different directions. So a heavy chain 39, heavy chain 40 are, tend to sort of need that 106 mutation first. But they seem to be somewhat incompatible with another light chain 109 mutation. So you can kind of get this different progression of the cells through the germinal center. This light chain 114 that comes up on day 7, the 106, and then different directions thereafter with different mutations coming through. Okay, so then could we identify pre-plasma cells in our populations? So we, when we got the UMAP, we sort of get this this old lady casting a spell on something, and what we could infer was, what we could see is this is the light chain population, the memory here. We had this sort of, when we had these within this light chain, we divide it into, light zone, we have these 3 different populations: light zone 1, which looks like sort of a canonical light zone population; 2, which seems to be becoming a memory cell population; and these LZ3 cells, which kind of tantalizingly lean towards the plasma cell population. And sure enough, they're enriched for cells that for genes that had previously been inferred to be pre-plasma cell gene markers. And if you look at those kind of that kind of signature, we compared it to a signature published previously by a group in Japan who had shown had identified cells they called fraction 1 cells, and those cells are characterized by the loss of BCL6, a beginning of the upregulation of IRF4, and actually an upregulation of CD40 and ICAM1. And sure enough, we're able to show that our LZ3 cells very much look like these fraction 1 cells. And more than that, by using the surface staining, the site-seq approach, we're able to see that these cells had begun to upregulate CD69. And finally, we were able, based on the fact that they expressed more CD40 ligand than ICAM1, we were able to sort these cells and actually show that they preferentially differentiated into plasma cells in vitro. So the question then was, do they have higher affinity cells than all the other cells in the germinal center? Are they therefore enriched for all these affinity increasing mutations that we've identified in the phylogenetic analysis? And the answer, perhaps surprisingly, was no. So this is sort of looking at the cells that have acquired, the darker color bars as opposed to the lighter color bars indicate that that the cell has acquired mutations at day 7. That's that L114F mutation. And you can see that these light zone 3 cells have no more of those mutations than any other dark zone or light zone 1 cells. Interesting, LZ2, so those are our sort of the things that seem to be heading off in the memory direction. They're actually less mutated overall. So that's very consistent with work from the nuisance group that suggests that memory cells come from low affinity cells. To do that statistically, what we were then able to do, because we've got multiple mice, was to essentially, we performed a logistic regression, basically asking what's the probability of having this mutation if I am a particular cell type. And formally, that's able to show that really these LZ3, these pre-plasma cell types, don't look any more mutated. In fact, they look rather less mutated than some of the cells that seem to be proliferating rapidly in the dark zones. The dark zones indicated in the sort of green color here. That's true at day 7, but that's very early on in the germinal center. But at day 21, we get the same pattern. So here, everything has now acquired this L114 mutation. So now we're looking at the second mutation, this Q106 mutation. And again, same thing. So we see very strongly that these LZ2 cells don't have this mutation, but the LZ3 cells do, but they certainly don't have it to a greater degree than the cells that seem to be continuing to recycle through the germinal center. And so from this, we kind of concluded that this early selection into the plasma cell compartment is affinity independent. And there are a number of other papers from the Nussensweig lab and from Oliver Bannard around this time, which sort of came reassuringly to a similar conclusion. And they were using that S1PR2 fate mapping mouse that we heard about earlier to sort of show that plasma cells and germinal center cells formed contemporaneously essentially have the same affinity. Just in the last few minutes, a couple of questions. So one question we had is there an avidity effect due to having CSP arrayed on the surface of the sporozoite? And it's in the paper, so I won't go into it. So no, we get the same effect with we're using a truncated recombinant CSP. Another question, though, was, of course, we've actually taken the whole spleen and we've mashed it up. We haven't looked at individual germinal centers. And we kind of asked, could we use a tree-based approach to try and identify cells that if they weren't in the same germinal center, were they at least in the same lineage? And finally, I guess, could it be that those pre-plasma cells had differentiated the previous day and were in fact high affinity cells, but we had just but we're looking at yesterday's germinal center rather than today's germinal center. So we're comparing apples and oranges, things that are mismapped temporally. So we made these kind of trees from each mouse, and because these are all, because of course we've put in our 2A10 cells, we get these very large trees. And what we've done is circled where particular important mutations arise. You can see that this L114 arises very early on, as you would expect. And then the 106 mutation actually arises recurrently through the tree at different points, but again sweeps through large parts of it. And then the other mutations arise later. The heavy chain, so at the time we did this, we couldn't do the analysis pairing of the light and the heavy chain, or we ran out of time basically because we were trying to get this story out because the other stories were coming. I now understand that in Dowser you can actually now combine the heavy and light chain analysis. And again, here you can see each particular branch of the tree is kind of dominated by one or other of these different heavy chain mutations that we saw early on. Another thing we tried that might be of interest was we had this idea that perhaps we could distinguish related lineages by their pattern of synonymous mutation. We sort of thought, oh, well, if things share synonymous mutations, they probably came from a similar common ancestor. Actually, it turns out that just as there's a canonical pattern of non-synonymous mutation, probably because of biases in AID and so on, you actually get a very canonical pattern of synonymous mutation as well. But nonetheless, if you look at the tree depth, there's essentially, well, a number of points. So firstly, the pre-plasma cells are scattered throughout the tree in sort of sublineages that have light zone cells and dark zone cells. So they're not all clustered in one place. So we assume they're coming out of multiple germinal centers, and they seem to be they seem to have no greater tree depth than their closest relations. And overall, both the light zone 3, the pre-plasma cells, and the memory cell and the pre-memory cells, they have as many mutations as the other cells, but they don't have the key affinity increasing mutation. So it's not that they are unmutated or they're yesterday's cells, they look like they're today's cells. So with that, I think I'm a bit out of time, so I will finish and just acknowledge the people that really did the work, Harry, Andy, and Haley, and a lot of support from Brian Parker and Teresa Neiman for the statistical side of things. So thank you very much.
Thank you. We have one question on the chat. Are the trees a lineage of a single clone or a combination of many clones?
Yes. So yeah, you're right. So essentially because it's a knock-in mouse, they'll all be the same. They'll all have the same common ancestor, right? But there'll actually be multiple different clones taking place in multiple different germinal centers.